Okay, I think it's time to get started. I'm sure we'll have more people join us in the next few minutes. Before we jump into the usual start of lecture, I have a few things I want to say. First is Canvas was accidentally set up to release the quiz 2 grades before we finalized them. So I don't know if people were getting sort of notifications that things were changing or if you had a look at them, but they haven't been finalized yet. So just keep that in mind. I think I've hidden them now, but hopefully we'll be able to release those by tomorrow. We're also going to be posting example answers for the short answer questions for those after the grades have been released. So you'll be able to look those over and compare your answers with the example answers. The other thing is the next online labs start today. When you come to lab, please remember to, before you click the link to join the lab, to use the SFU Zoom login. What that does is it associates your SFU email with your account, and I've preloaded all of the lab groups into the Zoom meeting, so it automatically assigns you to your correct group. If you don't sign in first, I have to manually add you in at the start of the class, so it just makes it a lot easier for me if you go through that two-step process before you come to the labs. Any questions about that? Okay, I also wanted to show you that I am participating in the pet plant project as well. Here's my plant. I'm growing radishes, so I'll keep you guys informed in how it's going. Okay, so I also love the annotations, the coloring today, a very happy looking moss sporophyte there. But unfortunately they have to go. Oh no. Okay, so last class, we were talking about the liverworts. As usual, we're going to start off today with a couple of questions based on material from last class. As before, our new protocol is don't answer the question until I tell you go. All right, here's the first question. Okay, you can go. Yeah, so it looks like people are in agreement here, and you are correct. So liverwort gametophytes are unisexual, which means that you have a gametophyte that is either male or female and will produce either antheridia or archegonia. The opposite then would be bisexual, which would be you'd have one gametophyte that could have both structures. Okay. One more question. Again, wait until I say go. Okay, you can start. So it seems like we've got B and C, there's a consensus. So B are the gemmae, those are the little sort of fragments of the gametophyte that can be dispersed, and they live in that cup, which is called a gemma cup. And everyone is agreeing that all three structures are haploid, which is correct. It seems like the harder one is A. So I think technically gametophyte and thallus are both correct, but thallus is more specific because sort of all three structures could be considered part of the gametophyte. A is pointing at sort of the main structure of the gametophyte, which in the hornworts that we've been talking about is the thallus. That's the thalloid gametophytes that have that sort of flat, sort of undifferentiated structure. So in this case, thallus is more specific, so that's what the answer is there, though gametophyte is also correct. And then we've got gemma cups, and then they're all haploid. Does anyone have any questions about either of these questions? Okay, so last class we ended up with a discussion of model species. And you guys did a great job coming up with a list of characteristics that researchers might look for when choosing a model species to look at. Everything from how easy is it to grow in the lab, what's the life cycle like, what tools are available, what questions does it help them answer. And I promised that we'd continue this discussion today. So what I really want you to do is I'm going to put you in breakout groups, and I want you to have a discussion considering all these sorts of different characteristics of model species. What are the pros and cons of using model species in research? So I want you to take some time, talk in your groups, and then in your groups, choose one person who's going to do the annotations when they come back. And I think it'll help keep the space manageable on the screen when you do the annotations. Does that make sense to everybody? Any questions before I send you away? Okay, just bear with me while I set up the breakout rooms. Okay, you should get the invitation now, and I'll see you in about 5 minutes. Okay, everybody should be filing back in now. So whenever you're ready, if whoever was decided would sort of report back in the annotations, feel free to begin. And if you didn't decide on someone, just go ahead and put your thoughts onto the slide. So we're getting some pros, large sample sizes with the specific life cycle considerations, I'm guessing. Inexpensive, fairly cheap and easy, well-known species, a base of comparison, applicable to many species, fast generation time. On the con side, you might not be studying the exact species you're interested in, won't have some sort of special features you might be interested in. You could be looking at some less complex systems, harder to study complex states. Lab results do not equal real life, could be oversimplified in the model. So lots of stuff about complexity there. Might be too hardy. Yeah, if you're picking something that's easy to grow and you're trying to see what the sort of effects are of manipulating things on its growth, you might get a misrepresentation of sort of a standard plant. Yeah, I think you guys have touched on a lot of the sort of the things that I was thinking about. I think it sort of fits in a bit on the pros is that one of the benefits of model species is that you can like hit a critical mass of people working on a species, which means that you can all feed off each other and learn from the research of other people. That lets you advance further than if you were just working on another species on your own, which relates to some of the stuff that you guys were talking about. Easier and cheaper. Often people pick species that because they're really reliable, so it takes some of the risks away of doing research if you can depend that your plants are going to grow every time and that your experiments are going to work, at least as far as, you know, keeping your plants alive. One of the other things is because there's lots of people working on something, you can develop common protocols, which means you don't have to start from scratch anytime you want to do something, which again comes back to building off of other people's research. As far as the cons, you've got some really good ones there. You're looking at one particular plant, so it might not let you, or one particular species, it might not let you ask specific questions or answer specific questions. One of the other things that I don't think anyone mentioned is it gives you a really biased view of diversity. Like you're sort of missing out on huge groups of species because you're focusing on a narrow set. And sort of if that model species doesn't do a good job of representing like the sort of other species, then you might have a completely biased understanding of what's sort of normal for plants or for your group in particular. One of the other things is that often model species are not, it sort of takes you further away from what might be called applicable research. If you're studying a model, like the classic model plant is Arabidopsis, which is like a little weed, and it's hard to justify to people, to taxpayers, the value of your research because it's multiple steps away from the possible applications of that research. And one other thing that I think is worth considering is who is making the decisions about what the model species should be, because realistically, that's very biased. Most of sort of modern science is conducted by white Western researchers, and they're the ones that are deciding what species are going to be studied. And so you're not getting the same sort of input and study of species from other parts of the world or that other types of people might be interested in. And so that's something to consider as well. Any questions about this? Anything anyone wants to add? Okay, let me just clear these. Another question someone asked last class was about any local liverworts. So I turned to my book that I showed you guys last class, The Plants of Coastal British Columbia, and I pulled up some fun looking examples for you guys. All of these are found in our sort of local area. And they're pretty, it's really interesting to see the diversity here, right? Because most of the stuff we've been talking about so far, it looks more like some of these phalloid liverworts, but when you see the diversity in color and the sort of the growth form of some like the leafy liverworts, it's really quite beautiful. I really like this purple worm liverwort, and not just because it's called a purple worm liverwort. So yeah, so I thought I'd share that with you guys as well. A liverwort salad, forbidden gummies. Excellent. So I'm going to try and sort of insert some local flavor into the lectures so that you guys can sort of see what's present around here. Okay, so that was the liverworts. We're going to move on to the remaining two groups of the bryophytes, the hornworts and the mosses. As promised, our learning objectives are the same as last time. We're just going to move on to the other plant groups. First, starting with the hornworts, which is the example of our background picture for today's lecture. So hornworts have a thalloid-like gametophyte, the same as the thalloid liverworts, but their sporophyte is this horn-like structure growing out of the gametophyte. And so this is shows you the scale. It's still a pretty small plant here, and a diagram showing what that looks like. And in this case, the hornwort, the sporophyte does have chloroplasts, so it can photosynthesize, which means that it can provide some of its own food, but it still depends on the gametophyte for resources as well. In this case, it's hornworts are one of the classic signs of hornworts is that they tend to have one single massive chloroplast in each cell, whereas the liverworts and mosses would be more familiar to you as having multiple chloroplasts in each cell. Like the liverworts and the other bryophytes, hornworts also have various adaptations to help regulate water loss. In this case, the hornworts actually have stomata, which is the first time we've seen that in the liverwort, in the bryophytes. And so these are the guard cells with the pores in the middle on the sporophyte. This is also where we're seeing some cuticle on the epidermis to reduce evaporation from the plant. And you can see underneath those, these are our stomata here. Underneath those stomata, we have intercellular spaces, these spaces between the cells that help to facilitate regulation of gas exchange and water evaporation, transpiration. Oh, I just noticed this looks like a little happy face inside the stomata. It's happy to see us. Okay, the other thing I wanted to mention here is hornworts also secrete this pectin-rich mucilage. You might recall pectin is the cell wall component that absorbs water really well. And so when the plant secretes pectin into the lower surface in these canals, it helps them absorb and retain moisture. It's also where the hornworts house symbiotic bacteria, which we talked about when we were first introducing the bryophytes. A lot of the bryophyte species in the liverworts, hornworts, and mosses have symbiotic bacteria to fix nitrogen for them. Pectin-rich mucilage is a good band name. No one would be able to spell it unless they had taken this class, though. Okay, so a little bit into the reproduction. So hornwort, antheridia, and archegonia are a bit different from the liverworts. They're actually sunken into the upper surface of the gametophyte. So in the liverworts, you'll recall that the gametophyte made these sort of protruding tree-like structures that would have the archegonia and antheridia, called the archegonia-4s and antheridia-4s, and those structures aren't present in the hornworts. They're literally just sort of in the surface of the gametophyte. Unlike the liverworts as well, the hornworts can be either unisexual or bisexual. So some species will have both antheridia and archegonia on their surface. In the case of the bisexual species, though, the antheridia will often occur before the archegonium development. And I have a discussion question here. Why do you think it might be advantageous for the species to have the antheridia development occur first? Okay, so I'm going to put you back in your breakout rooms and give you just a couple minutes this time to talk about this, and then you can come back. Any questions about, you know what you're doing? Okay. You should get the invitation now. There's a couple people who were late, so I'm going to assign you to some rooms. Okay, everybody should be coming back now. So go ahead and add in the annotations what reason do you think there might be for the antheridia to develop first? Any ideas? Energetically produce cheaper to make sperm cells, lower energy cost gamete, less energy demand, lower self-fertilization. So it seems like, to be produced before pollen has travel time. So it seems like a couple people are on the right track here. So the people that are talking about it being energetically cheaper to produce sperm than eggs, that is, I think, per sperm cell and per egg cell, that is true, but you have to keep in mind that the antheridium has tons of sperm. So I'm not sure if that's true, sort of broadly speaking, that an antheridium is cheaper than an archegonium to make. But a couple people talked about self-fertilization, which is on the right track. In bisexual species, you want to give yourself a chance for the egg to be fertilized by sperm from a different individual, because that's the whole benefit of sexual reproduction, right? And so by having the antheridium develop first and have that timing difference between when the two form, you are increasing the chances that self-fertilization will happen. It still gives you like some chance that self-fertilization will occur. Sorry, did I say that wrong? It increases the chances that self-fertilization won't happen and you'll get outcrossing between two different individuals. If necessary, like if there's no one else, no other plant nearby that can fertilize your eggs, there's still a chance that you can have self-fertilization and continue the next generation, but it just gives you better opportunities for outcrossing. Does that make sense? Any questions about that? Okay, it was a tricky one. So let's talk a little more about the sporophyte. So after the egg gets fertilized, like in the liverworts, the sporophyte grows sort of within the gametophyte. As we mentioned, you get the sort of horn-like structure growing out of the gametophyte. And it's actually will grow continuously, barring sort of other limiting factors. And that's because there's a region at the bottom of the sporophyte where the meristem is. And the meristem, if you're familiar with that term, it's sort of, we mentioned it briefly before, it's undifferentiated cells that will divide forever, basically. And so at the bottom of the sporophyte, there's this region that's going to keep making more sporophyte and push it out the top. And that's really beneficial because it lets it continually make more spores over time. Below the meristem, you have the region called the foot, which is again similar to the terminology that we talked about in the liverworts, as well as the placenta, which is gametophytic tissue that feeds the sporophyte. So even though in hornworts the sporophyte can photosynthesize, they still are dependent on the gametophyte for some of their needs, their nutritional energy and food needs. Yes, someone asked, outcrossing is the main advantage. Yeah, so when you've got the bisexual species, that timing difference is mainly to increase the chances of outcrossing. Yeah. Any other questions or anything you want me to explain one more time? Okay, so this is sort of the base of this, is the placenta 2N. No, this placenta, like in the hornwort, in the liverworts, the placenta is gametophytic, so it is haploid. Yeah. Okay, so let's look at the top of the sporophyte now. So as you move from the bottom to the top, the spores are developing as you sort of go through that vertically up, and you have this central strand, which you can also see here in this cross section, and it's surrounded by spores. And then at the very top, that's where the sporophyte will start to split open to release the spores. Hornworts don't have the elaters like liverworts do, but they have structures that have a similar function, so they're called pseudo-elaters. And like the elaters, they flex and move with the changes in humidity, and they help to catapult the spores further away from the sporophyte to disperse them. So I have a question here. So if you look at these spores, you might notice that this one, for example, it looks like there's 3 cells here based on the plane of section, but the spore itself, there's actually a 4th one on the bottom, and this is called a tetrad. And so I'm wondering if anyone knows, and we'll post in the chat, why do we see 4 spores in the development of the sporophyte? Any ideas? Yeah, it's the product of meiosis. Exactly. I've just got a slide to illustrate that to everybody. So meiosis is occurring in the sporophyte to produce the spores, right? You have the preliminary cell, which is diploid, and then it undergoes meiosis. You might recall that there's 2 rounds of division in meiosis, where a diploid cell gives rise to 4 haploid daughter cells. And so at a certain stage in the development of the spores, you have a structure that where you can see the 4 tetrads. And as the spore continues to mature, the walls between those tetrads will separate, and then you'll end up with individual spores. Any questions about that or any of the other hornwort stuff we've talked about so far? Unfortunately, there's no hornworts listed in my book, so I either there's none present or they didn't care about them enough to include them. So, okay, let's move on to the mosses then. So I think people are generally more familiar with mosses than the hornworts. They can have different forms though. You're probably more familiar with the upright variety, which are the ones on the left. But they can also have a feathery sort of form, more like flattened leaves or even sort of draping down. Again, all these 4 ones are all found in our area, so you can keep your eye out for them. Some of the mosses can also be epiphytic. Does anyone know what an epiphyte is? You can post in the chat. Any ideas? Grows on trees. Yeah, so they can grow on trees, but basically anywhere where there's not soil. Yeah, so like this example here, it's growing on a tree and sort of draping down. Yeah, so mosses can be epiphytic. Liverworts can as well, I believe. Some of the pictures that I showed earlier have some epiphytic liverworts. So let's talk a bit more about sort of the life cycle of the mosses. So here we've got a picture of a moss spore from which is growing a part of a structure that's called the protonema. The protonema is sort of like a network of single cells, and they can branch, as you can see here, and sort of form the start of the gametophyte. In this picture here, we're seeing the protonema in the sort of more transparent green. And then below that, the brown structure, the darker structure, is the rhizoid. So that's the sort of root-like anchor for the plant. Eventually, the protonema will get to a certain age and a certain stage, and they'll start to make a gametophyte bud, which is this little guy here. And as that continues to grow, you'll get a tiny little gametophyte that looks like this, that has sort of leaf-like structures and looks maybe more familiar as a plant. The leaves of moss aren't actually leaves because they don't contain vasculature. They are a single cell layer thick for the most part. Except for some moss have a midrib structure that can be multiple cell layers thick. And you can probably guess based on the color that these are their main function is photosynthesis. They've got a lot of chloroplasts and chlorophyll in order to make energy for the plant. This is a figure from a paper from a couple years ago, actually, that was looking at the genes involved in giving land plants 3-dimensional shape. It was really cool, but it was a little beyond the scope of the course. So that's all I took from there. So even though moss are non-vascular plants, they do actually have sort of simple rudimentary conductive tissues, but these tissues don't have any lignified secondary cell walls. Because they're not the same as vasculature, we use different terminology to talk about them, even though they might have similar functions to the tissues we've already mentioned. So for example, in blue, I've labeled what the tissue or the cells that are called hydroids, which you might intuit are water conducting cells. Like xylem, the water conducting cells are dead at maturity, but again, they don't have the supported secondary cell walls. This is a longitudinal section showing the same thing. And this is a cross section through one of the leaves of the gametophyte. So this actually the midrib part. So on either side, that would be a single cell layer thick, and here's multiple cell layers. And then in the center, you have the hydroids here. The other cell type here are the leptoids. So these would have similar functions as to phloem. So they're also called food conducting cells. And similar to the phloem sieve elements. But again, they're not the same as phloem. They evolved independently. Some mosses, not all, also have some supportive cells called sterids. And you can see they've got these thicker cell walls, but again, they're not secondary cell walls. It's just primary cell walls. So it does provide some structural support the same way that the thicker walls in Calenchima might, but they're not the same as fibers, for example. Does that make sense to everybody? Any questions? Okay. So let's turn to moss reproduction. So like the liverwort, are the mosses most closely related to vascular plants? So we tend to they have more things in common with the vascular plants, but I think if you look at I'm not going to go back to it because it's a long ways back now, but our sort of summary tree of all the major plant groups, it's not fully resolved, the sort of bryophyte branches. So I don't want to sort of make a claim, but they have a lot more in common with the vascular plants than the liverworts and the hornworts. They have more of the similar derived traits. Yeah. Okay. Yes, so like the liverworts and the hornworts, we have similar sort of terminology here. We have antheridia that make sperm, and we have archegonia that make eggs. In the case of the mosses, they form at the tip of a stem or a branch usually. So here we're looking at top down of one of the little gametophytes. So let me just go back. So once this guy's mature, it might have antheridia or archegonia in that little top bit there. The antheridia are on this, form sort of a splash cup to disperse the sperm. And the archegonia have a very similar structure to what we've seen before. You have the egg, and then you have the surrounding archegonial walls and these neck structures with a central canal through which the sperm will swim. Like the hornworts, again, we have some species that are unisexual and others that are bisexual. I mentioned the splash cup. Does anyone remember where else we've seen splash cups? Liverworts, yeah, exactly. If you remember the sort of like umbrella-like structure of the liverwort antheridia that acts as a splash cup as well. What is the point of the neck and the canal? So it's sort of a compromise. You want to protect the egg, but you also want to get the sperm access to the egg. And so it sort of gives an opening through which the sperm can enter, but it keeps other things away from the egg. Yeah. Interspersed in both the antheridia and the archegonia, sort of among them, you have these hair structures called periphyses. They're non-reproductive. So they, again, are sort of more protective to sort of prevent anything from sort of damaging the antheridia or archegonia. Could I point at the paraphil? Oh, the periphyses. So in this one, for example, there's in here, this sort of structure here, the hairs, the little hairs in between the different archegonia. And similarly in the antheridia, there's sort of hair structures. They're maybe a little easier to see in the antheridia because the archegonia are already kind of elongated, but the antheridia are more sort of swollen. So like this hair here is a periphysis. Are the neck and canal non-reproductive as well? Yeah, so we tend to say like sterile for non-reproductive. So in this case, it's sometimes called like the sterile jacket layer for the Antheridia, for example. Yeah, so they're not involved in the reproductive process except as sort of guiding and housing it. Okay. Oh, the closed captioning. Well, I'm actually pretty impressed with the closed captioning. So I'm impressed that it only got paraphysis wrong. Let's see if oh. Anyway, okay, we'll just ignore that. Singular of paraphyses. So I think it's paraphysis. Yes, I think it's a paraphysis. Okay. Any other questions about this reproductive phase? It's similar, like we've got water being involved here, the sperm are swimming to reach the egg, fertilization is occurring. What are the functions of paraphyses? So they are sort of protective of the Antheridia and Archegonia. If you imagine you just have the Antheridia exposed on a surface without the sort of supportive, like physically supportive, but also kind of keeping them separate from each other, keeping things from like bursting them before things are ready, just sort of containing everything. Yeah. Okay, so let's talk about what happens after fertilization. So here we've got the little leafy gametophyte, and the sporophyte is growing out the top. And at the top you have a little capsule and then a structure called a calyptra. Like in the hornworts, the moss sporophyte does have chlorophyll and can photosynthesize. So that's why it looks green. Here's a close-up view of the top of the sporophyte with the capsule at the bottom and the calyptra at the top. And it helps to protect the capsule, which contains the spores. In fact, the calyptra is actually part of the former Archegonium wall. This is a picture of a young sporophyte, and you can see the gametophyte is sort of stretched over the sporophyte as it grows, and that will eventually release and leave behind the calyptra at the top. So that means that the calyptra is actually, yeah, so the calyptra is one end, yeah, so the calyptra is haploid and is part of the gametophyte, and the capsule is a sporophyte. Here we're looking at a cross-section of the capsule. So there's this central structure here called the columella that has the tissue that makes the spores that undergoes meiosis, and the spores are stained red here on either side of that. As it grows, you'll eventually end up with a structure that looks more like this. Here, I'm just showing the stomata, because like the hornworts, the mosses have stomata. This is a zoom in of one of the stomata. It's sort of sunken into the tissue a bit. So these cells surround it, but then the guard cells are actually on the inside here. But you can see them sort of arranged, these little dots on the surface of the capsule are a whole bunch of stomata and guard cells. So this is a video showing the dispersal of spores from the capsule. I'm just going to let it play for a bit while I talk about some of this stuff, but we'll come back to that. Alien. Yes. Okay, so once the capsule is ready, the spores are mature, they need to sort of disperse from the capsule. Firstly, so this is separate from the calyptra. There's another structure called the operculum, and that is part of the sporophyte. So that's diploid. When the spores are mature, that pops off and exposes structures underneath called peristome teeth. And that's what we're seeing here, sort of bending back and away from the center of the capsule. And this is an SEM image, a scanning electron microscopy image of the peristome teeth. And you can see the spores in there too. So the cool thing about peristome teeth is that that opening of the teeth occurs as the cells dry out and the wall starts to flex, and it continue to move and sort of sway as the humidity changes, which is helping to release the spores from the sporophyte. Snails also have an operculum to hide in their shell. Oh, that's interesting. It's interesting to me how often because biologists are pulling from Greek and Latin to name different structures, sometimes you end up with similar terminology that might be related, sort of. I mean, you could say that they evolved together, or they might have arisen completely independently, and people working on mosses and snails were doing different things. Any other questions about this guy? Okay. This video, by the way, I took from Masaki Shimamura's YouTube page. She's got a lot of really cool videos. So if you want to check out more, I think I'm pulling more later on, but there's some fun stuff there. Okay. So that's the sort of last of the, oh, just to make sure the operculum is 2N. Yes, the operculum is part of the sporophyte. So it is diploid. Okay. So I wanted to end here by talking about a different sort of member of the general moss family. There's a whole bunch of different classes and groups within the mosses, and I didn't think it was worth sort of getting into the nomenclature of the different groups, but I wanted to talk a little bit about sphagnum mosses, which you might be familiar with as peat mosses. So sphagnum mosses are really important ecologically. They're a key part of building up sort of marshy, boggy ecosystems. So like ones that you see here. This is a picture of a sphagnum moss. Sphagnum mosses and peatlands in general are really important as carbon storage sources. There's some estimates that sphagnum mosses store more carbon than any other type of vegetation in the world combined. The reason that they're so good is that in the building of the peatland, you're actually sort of burying layers of vegetation underneath and having new layers on top. So it's literally sequestering the carbon that those plants have taken out of the atmosphere through photosynthesis and burying it so it isn't released again. There's a really awesome podcast from 99% Invisible that talks about this more. And it talks about the danger of, and in the past, people have gotten rid of peatland to plant forests because they thought forests were better for the environment than the peatlands. And now they're realizing how big a problem that was. And so there's people in that podcast, they were talking mainly in Scotland, I believe, that are trying to restore the peatlands. So I encourage you to check that out. There's some other sources here as well if you want to learn more about the peatlands and climate change. A couple other sort of interesting things about sphagnum. So sphagnum actually has a really interesting ability to absorb tons of liquid. And that's because they have these special cells called hyaline cells. They are like the sort of xylem vessels or the hydroids. They are dead at maturity, which lets them hold huge amounts of water. So even when you've like dried out the sphagnum, if you soak it, it's like a sponge. It can just suck everything up. In between the hyaline cells, you have these little narrow cells. That's not actually cell wall. Those are photosynthetic cells. So you can see most of the area of the sphagnum leaf is just these cells that will hold water. And the sort of value of holding water was not lost on people in the past. This is a vial of sphagnum moss that was used in surgical dressings in World War I. Because it could absorb a lot of material, and also because sphagnum makes peat bogs, like part of their biology is they make them acidic. And that acidic nature is actually helps prevent bacteria from growing. And so it's valuable in addressing not just because it's absorbing things, but because it's deterring bacterial growth. Oh yeah, so India was talking in the comments about bog bodies. Because they're acidic, they've been really interesting. Like people find, there was some, I think somewhere in the UK, they found a body in a bog and it looked so fresh that they called the police, even though it was like thousands of years old. It's fascinating. Someone also mentioned in the chat the sort of degradation of the peatlands. Part of that is that for in a lot of places, peat is harvested and burned as a fuel source, and that continues to happen to this day. And it's not classified as a renewable source of fuel because it takes so long for peat bogs to form, and not all peat bogs are continuing to grow. So it's continuing to be an issue in parts of the world. Any questions or anything anyone wants to add about sphagnum or peat? Peatlands also store a third of the world's carbon, but yeah, they're like a small portion of the land. They do amazing carbon storage, but we didn't appreciate that until fairly recently. Okay. We've got lots of good comments in the chat, and there was a link there as well. So I encourage people to check that out if they want to learn more about sphagnum. Okay, I've got some questions. Some of these will be pretty easy because you guys were asking me about this. So wait a bit until I tell you to go. Okay, you can start. Yeah, so this one, you guys are really like on to me in the types of questions I asked because you are correct here. A is the calyptra, which is haploid. That's a remnant of the Archegonia wall. And then the rest are all part of the sporophyte. They're all diploid. Good job. You're learning all my tricks. Okay. So we've got, because this is sort of the end of the sort of our survey of the bryophytes, I wanted to have a little bit of a summary. Any last questions about any of the sort of bryophyte stuff we've talked about so far before we move on? Okay, we'll just move on. If you have more questions, we can cover them later. So I want to have another discussion. I'm going to send you into breakout groups. I want you to think about how the different groups are dispersing their spores and what some of the similarities are in the strategies that they're employing in doing that. Okay, so that's the spore dispersal strategies among the three different groups and what their similarities are. So I'll give you 5 minutes to do that, and you should get the invitation now. Okay, the last few people should be coming in. So go ahead and write your ideas about what you talked about in your group on the slide. What are the similarities in spore dispersal strategy? Any ideas? Oh, so we've got a good one to start with. Elaters, pseudoelaters, and peristome teeth all flex with humidity. That's a good one to help disperse the spores. react to moisture, need water. Splash cups were similar between the mosses and the liverworts. Yeah, it seems like most of you got that part. Tall, elevated structures for spore dispersal. That was the other main one I was looking for. If you think about the archegoniophore in the liverworts, you've got the sort of tall structure. With the hornworts, you have the top, they disperse from, the top of the horn. And with mosses, again, you're making that tall capsule structure on the end of a stalk. So that helps the spores to disperse as well. Air or water for spore dispersal? It's mostly air, I would say. The water would work too, I guess, especially if you've got sort of rain happening. It might be that I'm biased because you don't take pictures when the moss is underwater or whatever. But yes, yeah, excellent job, everybody. Any questions about this? I wanted to include this question because I find that often we pay so much attention to the differences between different plant groups that we lose track of the things that are in common. So that's just something to keep in mind when you're studying as well. Okay, so I have one last sort of summary activity. In the pre-class slides, you'll find this table if you need to refer to it, but basically I'm going to put you back in your groups for 5 minutes or so and see what you can fill out in this table comparing the different species we've talked about. Okay? Is anyone writing this down, or you can find it in the pre-class slides if you need to refer back to it? Okay. So I will put you in your breakout rooms again, and I'll see you in a couple minutes. Okay, the last few people should be coming back into the main room. So maybe let's start with the first column. So this is how dependent is the sporophyte? You can go ahead and put your thoughts in for each of the liverwort, the hornwort, and the moss. Yeah, so we've got, it seems like everyone's saying the liverwort, the sporophyte is completely dependent because it's non-photosynthetic. With the hornwort and the moss, it's still dependent on the gametophyte. It's still, like for the hornwort, for example, we looked at the sort of placenta that the gametophyte makes to feed nutrients to the sporophyte. But because the sporophyte in the hornwort and the mosses have chloroplasts, they are not as dependent on the gametophyte as the liverworts are. Okay, what about the conducting tissue? What did you guys decide on? So it seems like for the moss, people are talking about the hydroids and the leptoids. The hydroids are the sort of water conducting cells that are analogous to xylem vessels. And the leptoids are the food conducting cells that are analogous to the sieve elements in vascular plants. Someone mentioned sterids, which are a specialized cell type, but they're not a conducting tissue. That's a structural support tissue. And liverworts and hornworts don't have any sort of rudimentary conducting tissue, unlike the mosses. So that's correct as well. Okay, next up, stomata. What did you guys decide on here? Oh, I like the green check and the red X. So Yes, so the liverworts don't have stomata, but hornworts and mosses do. And we looked at some on the moss sporophyte, for example, on the capsule. And someone mentioned in the liverwort that there are air pores, which let gases into the sort of intercellular spaces in the liverwort. So you're correct that it's sort of serving a similar function to the stomata. It is, I would consider, a notable trait. Any other notable traits or structures in each of the species that you guys came up with? Anything unique to each of the, yeah, the liverworts are unisexual, and the hornworts and the mosses can be either unisexual or bisexual. The liverworts can have gemmae in the gemma cups. The hornworts have sunken gametangia. Anything else? So some of the other things I came up with, so in the hornwort, they have that the sporophyte does that splitting thing to help release the spores from the top of the sporophyte. The mosses, we talked about the protonema, that network of filaments in the gametophyte before the sort of leafy part buds. Yeah, there's a whole bunch of things that might fit into that category. Okay, so I find this kind of exercise of sort of setting up tables to help you compare between the different groups really helpful. So you might want to maybe try exercises like this as we go through the different groups. That's all we've got for today. Any questions about anything we've covered today or generally in the bryophytes? Okay, so as usual, coming up, we've got next class, we're going to move on and start to look at some of the vascular plants. But before we get there, you guys have to make it through lab 3, where you're going to be exploring the bryophytes some more in some activities. And then the third quiz will open on Friday at noon. Are the stomata only present in the sporophyte of mosses? Yes, I believe that is the case. The gametophyte, if I go back to look at the leaf, they're only a single cell layer thick, so it doesn't make sense to have stomata on the leaves, for example. And similarly with the protonema, they're a single cell layer, so they don't have stomata either. But then on the sporophyte, it's sort of a big multicellular structure, so that's where you're going to get the stomata. Any other questions? Oops. Okay. Okay, so that's the end, and I will see those of you with lab today in a few hours, and the rest of you in the next couple of days. Bye, everybody.